This is the lecture for Lab 14 on evolution. The theory of evolution is an idea that incorporates many different ideas and also data from many different experiments. And a definition of evolution could be all the changes that transform life on Earth, leading to the diversity of organisms. Charles Darwin is considered the father of evolution, and he was a naturalist on a ship that sailed around the Galapagos. And he was responsible for taking samples of the organisms and animals that lived there and bringing them back to England. And as he observed the different types of finches and their different beak styles, he noticed that it matched what food was available on the different islands. And from this, he came up with the idea of evolution and natural selection. Populations are very important in the idea of evolution because populations are the unit of evolution. Only populations can evolve, not individuals. And a population is defined as a group of individuals of the same species living in the same place at the same time. The definition of this population means that it is a group of individuals that can interbreed and share genes. Natural selection is the mechanism of evolution. So this is what will cause populations to change over time. And for natural selection to occur, there are a couple of different requirements. First, that population has to have variation in their characteristics. Second, there must be a selective pressure, so that not all members of that population will be able to survive. Third, individuals with favorable traits will survive and they will reproduce. And fourth, as you go across generations, a larger portion of the population will possess the favorable traits and will become adapted to the environment, to that particular selective pressure. As an example of natural selection, let's say you have a valley, and in the valley you have a population of rabbits. So in that population of rabbits, the rabbits have different variations. There could be variation in color, there could be variation in how fast they run, how high they jump, could be variation in how well they hear, or how well they metabolize glucose. But the point of the population is that they are not all identical to each other. So that's your population with variation. Then a fox moves into the valley. So the fox is going to be the selective pressure. Now with this selective pressure, not all of these rabbits are going to survive. So whatever favorable characteristic enables the rabbit to survive will be passed on through the generations. So let's say that this rabbit runs faster than the other rabbit, then this rabbit is more likely to survive, and that rabbit will have offspring, and all of that rabbit's offspring will be faster. So over time, with the selective pressure, the average speed of the rabbits in the population will increase, because those that can run faster will survive, they will reproduce, and their offspring will also have the ability to run fast. There is a lot of different types of evidence for evolution. The most common evidence that you are familiar with are fossils. Fossils are remains or impressions of living things that are found in the earth and in the rocks. And by looking at the fossil record, we can see how species have changed over time. Another type of evidence is embryological evidence, and this is looking at structures that embryos have that the adult organism does not have. And if you look at humans, during our embryonic development, we have gill slits at one point, and we even have a tail. And that is a shared characteristic with other types of organisms like birds, reptiles, and fish. And that implies that we have a common ancestor. Homologous structures are also evidence of evolution. Homologous structures are structures that are found in closely related species but have very different uses. So humans Cats, whales, and bats are all mammals. They have similar bones that are found in the forelimb, but they all have very different structures. The human arm is used for grabbing, the cat's leg is used for walking, whale's fin is used for swimming, and the bat's wing is used for flying. Analogous structures are structures that you find in distantly related species, but they have the same function. So the wing in a bird and the wing in an insect have the same function, but the two types of organisms are very distantly related. Vestigial structures are also evidence of evolution. These are structures that no longer have a purpose in the current species. Some examples of vestigial structures in humans are wisdom teeth, ear muscles to move the ear, the tailbone at the bottom of the spine, and the appendix in the intestines. All of these are structures that do not have any purpose in the modern human. A cladogram can be used to arrange species by shared characteristics. To design a cladogram, you create a diagonal line to the right, and you have different characteristics listed on this line. And for a shared characteristic, everything that comes off of the line above the characteristic is shared. So for example, vertebrae. All of the species here have a vertebrae. Sharks, ray fin fish, amphibians, primates, rodents, crocodiles, and birds. Then as you go up to bony skeleton, all of them share a bony skeleton except for the sharks. Their skeleton is made of cartilage. Then as you go up to four limbs, all of them share four limbs except for the fish and the sharks. And it goes on so forth. So amniotic egg, all of those share an amniotic egg except for the amphibians. In this way, you can determine which organisms are distantly related and which ones are more closely related. For your lab report. For experiment 1 on the transformation, this is where you would have recorded the data that you would have generated from the previous week. These are the 3 plates you would have seen. Plate 1 would have had confluent growth, tons of growth on both sides, 1A and 1B. Plate 2 would have had no growth on 2A and would have had a few colonies in 2B. Each round white circle is a colony. On plate 3 in 3A, you would have seen no growth, and on the side 3B, you would have had colonies that have a blue color. So use that information for your lab and then answer the following questions. For experiment 2, homologous structures, you would have looked at a human skeleton and the skeleton of a cat. The human skeleton has certain bones that are numbered 1 through 5. So 1 is here, 2 is here, 3 is here, 4 is here, and 5 is down here. So you would have looked at those numbered bones, determine their name, and then determine on the cat which are the correlating bones. So A is here, B is here, C, D, and E. For the comparative anatomy for analogous structures, for this you would have looked at a chicken wing and a bat wing and answered the questions looking at those 2 structures. For vestigial structures, you would have looked at the forearm structure for the human and the frog and answered questions related to those diagrams. The last experiment is experiment 3, and this is the Cytochrome C Cladogram. So for this experiment, you are going to design a cladogram that will show the differences between species in terms of the Cytochrome C protein. So Cytochrome C is a protein that is found in many different species, but it has slightly different structures in the different species. So the species you are comparing are human, shark, horse, turtle, monkey, chicken, and frog. And for this, you are comparing different amino acids in the protein, and the number of the amino acids is listed there. The position number is listed there. So the first thing you are going to do is compare the human to the shark. And to compare the human to the shark, you are going to compare the 2 amino acids at each position. And whenever you find a difference, you are going to count that difference. And then the position 46 is different. This is same, different. So you'll go through the whole list and count up the differences between human and shark, and you record that number on the table. Once you have compared the human to the shark, then you compare human to the horse, human to the turtle, human to the monkey, human to the chicken, and human to the frog. Once you have recorded all of those differences, then you are going to compare the shark to the horse, shark to the turtle, shark to the monkey, shark to the chicken, and shark to the frog, until you've gone through and compared every species to each other species. And again, on the table, you are recording the number of amino acids that is different. Then, to make the cladogram, on your lab report, you already have the line that is angled up to the right, and you are comparing all the other species to humans. So humans are at the end. And for this, you are not comparing physical characteristics, you are comparing number of differences. So find the species that has the highest number of differences, and let's just say it's 20 differences in amino acids, and you write the name there. Then you find the next species that has the next highest number of differences, let's say it's 10, and you write the name there. If you come up with 2 species that have the same number of differences, let's say it's 5, then you create a fork and write the 2 names of the species there. So that's what you should produce for the Cytochrome C Cladogram.